Thank you for all coming along today. Of course, I'd like to start off by acknowledging the traditional owners of the land, the Wurundjeri people, and of the Kulin Nation, and pay my respects to their elders, past and present, and of course, any elders from other communities who may be here today. I first met Najoua at the very end of a TNF meeting. It was literally 5 minutes before the end when we were all leaving and I had my suitcase and I was walking out the door when she finally plucked up the courage to approach me about doing a postdoc. Her timing has always been impeccable. To those of you who know her vibrant personality, it's hard to imagine that shy PhD student, but there you go. And I'm glad she overcame her shyness then. At that time, she was doing her PhD in Dijon, France, with one of the early stars of the TNF signaling field, Olivier Michaud. And together with Jörg Chopp, he was the first to identify that second signaling complex downstream of TNF, which you are about to hear so much about. Many students in the audience may dream of getting a Nature article, as Najoua has done. And I thought, do I have a message for them? Well, the reality is this was an enormous effort and many, many nights and weekends from Najoua, and I think we have to acknowledge that. And it took time. I first talked about our quite advanced results at the end of December 2017, and so there was another 2 years of very hard work to get it to the stage where it is at. But I do have two other quick good news messages. One is that communication and sharing of our results at that very early stage in advance of publication was a great positive. And I think that Najoua will highlight some of the positives that came out of that. And it led to collaborations and a more impactful public story. And many of us are fearful of getting scooped, so we really need to acknowledge the times when it actually works to our advantage to do what we naturally want to do, and that's to share our results and talking about our exciting findings. And secondly, and I think this is really important, why I saved it to last, is that frankly, this work arose out of testing a very simple hypothesis. That influential hypothesis had been put forward in a couple of Nature papers in 2011, and it stated that RIP1 cleavage the purpose of RIP1 cleavage was to prevent initiation of necroptosis. And Najoua and I realized that despite its huge traction in the field, that this had not actually ever been tested. And the result of that testing is the story that Najoua is about to present, and therefore I'm very much looking forward to hearing about it in her words.
Thank you very much, John, for this introduction. So, yes, I am really, really excited today to present our latest discovery on RIPK1. And I will talk about its pathological role in human disease. And as John said, I will talk about the fact that it's not all about this hot and highly inflammatory type of cell death that is called necroptosis. But before, I'd like to really acknowledge and precise that this study is the result of a really fruitful collaboration with the group of Dan Kastner. So his group is at the NIH, and Dan is a world expert in the autoinflammatory disease, and together with his former postdoc, Stephen, that is now a lab head and hero, I couldn't find a picture, but it looks like him, really. So together with Stephen and Hero, we have discovered a new autoinflammatory disease that is driven by mutation in RIPK1. So I wanted to start off with some really simple background about why I'm still working on cell death since 15 years and how the field have shifted from to work on cell death and inflammation and how those processes are molecularly linked. So in a context of inflammatory diseases, the main feature is the production of inflammatory mediator, and these induce more inflammatory mediator that is due to the activation of signaling pathway that are the NF-kappa B and MAP kinase and others. And our favorite inflammatory mediator is TNF. We think that's the most important molecule in the world in our lab. And its importance in inflammation led 20 years ago a number of pharmaceuticals to develop those drugs that would block its activity, which we now call the biologic anti-TNF. And the success of those drugs was assumed to be due to the inhibition of TNF-kappa B MAP kinase and inhibition of production of more inflammatory mediators. However, TNF has another outcome, which is cell death. And in actual fact, the molecular signaling cascade that is responsible for this survival/inflammatory signals is inhibiting this cell death. So in most cases, TNF does not kill, it induces survival, inflammation, and proliferation. And however, a number of those knockouts that activate this survival pathway has showed us in the last decade that in actual fact they do induce cell death, but also one would think that if you shut down this arm, you would not have any more inflammation, but if you shut down this arm, you actually have inflammation. And that's where comes the concept of cell death driving the inflammation. And that's what a part of our field is trying to understand is how is this cell death activated, when, where, and by which cell type, and can we target it for inflammatory diseases, for example. And the other argument for us to be even more interested in trying to understand which type of cell death is from the discovery of necroptosis a decade ago, which is a highly inflammatory type of cell death. So TNF has the ability to activate either a caspase-dependent type of cell death called apoptosis. This is considered to be immunologically silent, and also a caspase-independent type of cell death called necroptosis, and this is highly considered to be highly inflammatory because this is a lytic form of cell death where there is the release of danger signal that would activate inflammation. However, our challenge is RIPK1. And the challenge for us to understand when does TNF switch from the survival to cell death is because some of the molecules that regulate all those arms, they play a role in every single arm. So RIPK1 is one of those. RIPK1 is believed to be needed to induce this survival and inflammation, but it also plays a role in necroptosis and apoptosis. But the other challenge about studying the role of RIPK1 is also because it regulates many other signaling pathways that are involved in immune responses. So now I will take you through the journey of RIPK1 in the TNF signaling that you can have an idea of how this molecule switched the TNF survival signal to a TNF cell death signal. So when TNF binds to its receptor TNFR1, there is the formation of a complex at the membrane, and RIPK1 gets recruited, and there is a number of E3 ligases that ubiquitinate RIPK1. And this ubiquitin platform is a docking site for the recruitment of kinases that are involved in the activation of the MAP kinase and the NF-kappa B pathway. And that's what is driving transcriptional response that is leading to survival, inflammation, and proliferation. And amongst those kinases that are recruited, some of them phosphorylate RIPK1, and I'll come back later to it. And that's what John said, and there is a secondary complex that has been discovered by my PhD supervisor, and what we understand today is that this complex is slowly internalized in the cytosol, and RIPK1 can bind to Fad, which bring caspase to the complex. And the transcriptional response from the complex one produce anti-apoptotic proteins such as C-flip, and C-flip is a caspase inhibitor, comes to that complex and kind of inhibit the caspase activity halfway in a way that it inhibits caspase to cleave caspase 3 and therefore inhibit apoptosis, but it allows caspase to cleave RIPK1. And this cleavage, as John said, is believed to inhibit RIPK1 to interact with RIPK3, and in the end, to inhibit activation of necroptosis. What is believed before we published is that RIPK1, when it's not cleaved, is interacting with RIP3, activating RIP3. RIP3 phosphorylate MLK in turn, MLK goes to the membrane, makes those spores, and then that's when you have these dangerous signal that gets out and presumably induce inflammation. So one would imagine that if you, not sure it's going to work, that thing. So one could imagine that if you inhibit any of these ligases or kinases, you have cell death happening. And something that we still don't understand, but this complex is rapidly formed and activated, and that's where the cells die, either through apoptosis, and if there is any defect in the caspase activity, you get necroptosis. So a couple of years ago, I was interested in more in cancer. I'm still interested in cancer, but most of my projects were around trying to find therapeutic strategy to kill cancer cells. And I was trying to manipulate the TNF signaling to switch from its survival mode to his cell death mode. And I took advantage of the clinical development of those drugs that were called SMAC mimetic. So SMAC mimetic, they inhibit the function of CIPs, and they are one of those E3 ligases that ubiquitin RIPK1. And if you treat cells with SMAC mimetic, RIPK1 is no longer ubiquitinated, and you have this rapid internalization and activation of cell death. However, there was a number of cancer cell lines that were not responding to SMAC mimetic, and that was one of my duties to understand what were the molecular mechanisms behind this resistance. And this is a long story short, from several studies from our lab and other labs, and collaboration with Pascal Meyer in England. But what we found in actual fact is that the MAP kinase MK2 was phosphorylating RIPK1, and this was limiting RIPK1 to get into that complex and limiting the death induced by SMAC mimetic. And I think yeah. And it was great to see that if we were treating cancer cell with a SMAC mimetic and an MK2 inhibitor, we can sensitize a number of cancer cell lines. But as I said, the only thing that we knew that was that MK2 was phosphorylating RIPK1, but we were not sure how this phosphorylation was making RIPK1 more accessible to that complex. And John was interesting to understand we were together interesting to understand what was the molecular mechanism explaining this new regulatory molecular event of RIPK1. And he looked at the sequence of RIPK1, and what he found is that the serine that are phosphorylated by MK2 are highly conserved. And interestingly, they were flanking the caspase cleavage side of RIPK1. So from there, we came up with several hypotheses, and we thought that maybe those two molecular events, phosphorylation and caspase cleavage, are talking to each other to regulate the function of RIPK1. And one of the hypotheses that we put forward was that the phosphorylation of RIPK1 by MK2 is increasing the cleavage of RIP1 to inhibit necroptosis. And to address this, we use the magic of the Magic Lab with Andrew and all his team with Marco. And we've decided to make RIPK1 mutant mice, CRISPR knock-in RIPK1 mutant mice. And the reason was because we wanted to go away from using an MK2 inhibitor, because MK2 is also another kinase that has a lot of function other than phosphorylating RIPK1. So we made a phosphosilent mutant where we replaced the serine recognized by MK2 to an alanine, so that would abrogate inhibit the ability of MK2 to phosphorylate RIPK1 and therefore mimic an MK2 inhibitor. And we replaced this highly conserved aspartate that was recognized by caspase to an alanine. And this should be producing a non-cleavable form of RIPK1. And our hypothesis was that if RIPK1 is no longer phosphorylated, there is no cleavage happening, and maybe we get spontaneous necroptosis, and we were thinking that we may get some sort of inflammatory phenotype in the mice. However, the mice were viable and healthy. And this was not really disputing our previous finding with the MK2 inhibitor, but it was just telling us that this phosphorylation event was a secondary checkpoint on RIPK1, a backup plan if the level of IRP was low, MK2 was there to be able to block the cytotoxic activity of RIPK1. And in fact, a PhD student in our lab, Denise Heckman, she tested cells from those mice, those phosphomutants, and she found that if they were treated with SMAC mimetic, they were highly sensitized when compared to the wild type. So that was confirming what we found, that MK2 phosphorylate RIPK1 to block his cytotoxic activity only when RIPK1 was not debutylated. So result from the uncleavable RIP1 was a bit more interesting though. What we found is that those mice were actually lethal. And the reason of this interesting finding was coming from the fact that loss of RIP1 in mice die at birth. So one would think that RIP1 may not have a role in embryonic genesis, and whether it gets cleaved or not would not matter. However, our mutant mice was dying at mid-gestation. So now that's where RIHI is amazing, because I have the honor of working with two experts in embryogenesis, and that was Anna Voss and Anne Voss and Christine Biben. And with Anna, we have analyzed the embryos and determined when they were dying. And what we found is at day 10 of embryogenesis, some of the embryo were showing mild to severe hemorrhage throughout the body. And by day 11, all the embryo were dead. So that was telling us that these embryos were dying at E10.5 and from vascular defect. And then the other expert was with Christine. So she was reanalyzed and took the embryo and analyzed the histology. And what she found is that those embryos at E10 were presenting with cardiac defect and neuronal retina defect. So those lethal features were strikingly similar to what is happening in the caspase and the FAD knockout. So caspase knockout and FAD knockout mice, they also die at E10 from vascularization and cardiac defect. And the reason for these mice to die, it's because there is a spontaneous overwhelming activation of the necroptosis. Because if FAD is absent, caspase is not brought to the complex to cleave RIP1. And obviously, if caspase is absent, RIP1 is not cleaved, and that's where is coming our current understanding how necroptosis is repressed. And RIP1 is not cleaved, and that's where you get necroptosis. And this was demonstrated genetically by different labs, and including the lab of Andrea Strasser, and the cross of caspase or FAD to RIP3 or MLKL were rescuing the lethality of caspase and FAD. So that was telling us that at E10, those embryos were dying from necroptosis. So then the question was whether our uncleavable RIP1 was also dying from necroptosis. And again, we are using the MAGI of the Magic Lab again, and that's where we are probably going to go back. To address this, we made a double mutant mice, and we killed the kinase activity of RIPK1 in our uncleavable RIP1. And the reason is because the kinase activity of RIPK1 is known to be involved in activating the necroptosis pathway. RIPK1 autophosphorylates, this induces phosphorylation of RIP3, and which leads to MLKL phosphorylation and MLKL to the membrane and release of DAMPs. And so we asked Andrew to make a mutation in the kinase domain of RIPK1 of our uncleavable mice, because we knew that mutation would kill the kinase activity. And this work was done with, in conjunction and collaboration with a group in Germany from Pasparakis lab that also made the mice in the lab. And what we found is that the uncleavable RIP1, when they have a killed kinase activity, they actually survive, but after a couple of days and weeks, they started to show an inflammatory phenotype. And first, we thought, okay, this is necroptosis. Our embryo, they are dying from necroptosis, but there is something else happening after. And here I'm showing pictures of those mice at almost 3 weeks old here, and you can see that this is their little mate. So they are really runted and have a typical inflammatory phenotype. And postdoc from Manolis lab has analyzed a number of organs from those mice, and what he found was a multi-organ inflammation, which was affecting the skin, the liver, the lung, and the spleen, and he also found some patchy Caspase-3 positive cells in some of the inflamed organ, which was telling us that cell death was happening in those mice. So now we are puzzled with John. We don't understand. We're still asking whether this is necroptosis. I am still asking whether those embryos are dying from necroptosis because of this particular phenotype that is happening. And John is really convinced this is necroptosis. We are arguing, as usual. We always argue about science, but that's what makes great science, right? So we bet a bottle of wine. And one of the other ways to determine whether necroptosis was killing those embryos was to cross to the latest known effector of necroptosis, which was MLKL. So we bet on that cross. So John is thinking it's rescuing. I'm thinking it's not rescuing. What do you think? This is not rescuing. So I was quite excited because I thought I'd get a really nice bottle of wine here. From the region where I'm from, Burgundy. But I got instead that bottle of wine that he got with his Quintas freaking friends. It was a really nice one. It was really nice. But I was expecting this. So MLKL is not rescuing. Those embryos are not dying through necroptosis, but the kinase activity is rescuing to birth 2 weeks. And even more surprisingly, if we cross to this other necroptotic effector, RIP3, we do not have a rescue, but we still have some sort of rescue to E12. So RIP3 is doing something to bring those embryos to E12. So what we've highlighted here with those crosses is a necroptotic role of RIP kinases during embryogenesis. And There is quite a few work out there that is suggesting that, in fact, the kinase activity of RIP1 is also involved in regulating apoptosis. We're quite not sure what are the molecular mechanisms. And similarly, for RIP3, there is some work showing that it can regulate apoptosis. So we thought, okay, these rescue from kinase activity of RIP1 and RIP3 are because this is inhibiting apoptosis. And that's what we did. We crossed the mice to caspasate that is involved in apoptosis. However, I mentioned that the caspasate knockouts are dying from necroptosis. So the only way to determine whether apoptosis was activated, we also had to get rid of RIP3. So we crossed the mice to RIP3 and caspasate, and that's where we had a nice rescue. And even more interesting, just one loss of one allele of caspasate was enough to rescue the lethality of the includable RIP1. So what we're showing here is that the cleavage of RIP1 is not solely to inhibit necroptosis, but also to inhibit apoptosis. And in actual fact, I think we haven't rigorously addressed whether necroptosis is happening here, because we had to knock out RIP3 in addition of caspasate to be able to see the effect of caspasate. So what is killing the embryo, what is inducing this cell death, which ligand? And we went for the obvious one, TNF, because this is where RIP1 plays its major role. And we made mouse embryonic fibroblasts out of our E10 embryo and looked for their sensitivity to TNF. And here I'm showing a PI-positive cells that would tell us how much the cells died, and measured by FACS after 24 hours of TNF treatment. And I was really not impressed about that. And the reason is not because I absolutely want to have the result I want, it's because when I was looking at my cells earlier, they were dying, I was objective, they were dying more. But what I thought after is, in actual fact, there is a lot of death happening in the beginning of the TNF treatment. But then the survival cells were proliferating, taking over, and we were underestimating the cell death happening here. And that's where WIHA is an amazing place, because I bumped someone else that helped me a lot in that project, which is who is Christy. So I explained her my existential problem. And she tells me that she just purchased this amazing machine that we all in the cell field are in love now, which is the InQSight. It's a time-lapse machine that is really easy to use and allow us to do billions of experiments at once and analyze it from your couch at home. And so we started together to set up, optimize the condition to be able to ensure that we are not missing the molecular event or the cell death happening in our cells. And what we found is that the MEFs from the E10 embryo were actually dying upon TNF. And this death was happening quite early. Here we are looking at, by the end, most of them are gone, and this death is happening quite rapidly. From the first hours, we already have 50% of the deaths, of the cells dead. Sorry. I need to hurry. So then we like to always understand what type of cell death is happening in our cells, and we have treated with a caspase inhibitor to know whether apoptosis was activated, and we could not rescue the cell death. So that was telling us that we have pushed the cells to necroptosis, and that means that it's not solely apoptosis that is happening in those cells. And treating with a RIP3 inhibitor, which should inhibit necroptosis, was also telling us that it's not only necroptosis happening in these cells. So we think that there is both cells happening in here. But if we delete RIP3 or kill the kinase activity of RIP1, we have a complete rescue of this death. So one would think here that this is necroptosis, and therefore a RIP3 inhibitor should inhibit, but is not doing the job. And similarly, kinase activity of RIP1 should inhibit necroptosis, which does it here, but the RIP3 inhibitor is not doing it. So what we're thinking here is that RIP3 scaffold function and RIP1 kinase activity is activating caspasate in that complex when RIP1 is no longer cleaved. And this is where it goes from my title, moving beyond necroptosis, is actually this cleavage is not solely to inhibit necroptosis, but also to inhibit caspasate. And we've highlighted a new role of RIP3 in that complex, and I will explain later what we think is happening. Now, while we are playing with our mice and cell death pathway and so on, another really nice thing at WeHi is Seth Master comes to us and he said, you know, there is Dan that has a cohort of patients that has a mutation on the cleavage site of RIP1. And we are extremely excited because we think, okay, we have the unique opportunity to be able to determine whether this mutation is causative of an autoinflammatory disease. And before I get on describing Dan's patient, I just want to touch on really on what are autoinflammatory disease. So they are not to be confused with autoimmune disease because they are due to a defect in the innate immunity. And unfortunately, those patients, they suffer from systemic inflammation. And one of the main characteristics of the monogenic autoinflammatory disease, which are driven by mutation on genes that regulate the innate immunity. So one of the main characteristics is periodic fevers. So they have fevers throughout their life, and this is not due to infection. This is due to an autoinflammation. And they also present with different clinical manifestations that are or rashes or big lymph node or liver and so on. So in 2010, Dan received Victoria. She comes with, she's 1 year old, and she had periodic fever since she's born. And she presented with other clinical manifestations that were typical from an autoinflammatory disease. And this is the beginning of the sequencing, whole genome sequencing, exome sequencing era in the beginning of the decade. And that's when Stephen sequenced the samples from Victoria, and he did a whole exome sequencing. And amongst the mutation that he found in Victoria's sample, he found this mutation in the caspase-1 cleavage site, but only in one allele. So he has done his homework because he looked at the role of RIPK1, and at the time we already knew that it has a role in regulating inflammation, and he proposed that to a lab meeting where he thinks that this mutation is driving the disease. However, he had to chase up for a second family to be able to be sure that this mutation was driving the disease. And that's where they went to old samples from a family that was followed by Dan for 20 years. And this family had 3 generations with affected individuals. And similar to Victoria, Stephen has sequenced the samples, and he also found a mutation on the RIPK1 cleavage site, but this time it was not an aspargine, but an histidine. And again, those patients were heterozygote. So then early last year, when we started to work together, that's when you feel like there is a lot of luck in life. We had a third family coming to Dan's service. This is Adam, he's 6, and the same, he's presenting with periodic fevers and enlarged lymph node and a number of other clinical features of autoinflammatory diseases. And Adam also has a mutation in the cleavage size of RIPK1, but this time this is a tyrosine. And Hero was great at the time because he managed to get Adam during a flare, just after a flare of his periodic fever, because in the 20 years, that was the difficulty that Dan was confronted to, is the fact that when he was seeing those patients, they were either under treatment or between flares, and he was not able to determine what kind of inflammatory marker they were having, and that could explain their condition. And so Hero is looking at the cytokine in the bloodstream of Adam, and what he found is that the inflammatory cytokine TNF, IL-6, and IL-1 were up when he compared to control. And even more interestingly, he also did some RNA-seq analysis on the blood of Adam, and he found inflammatory gene signature in his PBMCs. But we had to make sure that all those different mutations were also blocking the cleavage of RIP1. And that's where Hero has made a number of constructs that were expressing the mutation found in our patient, in their patient, and in our mice, because we used an alanine. And he did an in vitro caspase assay and to look whether caspase could cleave, and you can see here that caspase was not able to cleave any of those mutants. So any mutation of the aspartate would abolish RIPK1 cleavage. So from there, we needed to find a disease. We were quite sure that this mutation was causing the disease. And after lots of email during the middle of the night, we came up, I think that was Stephen and I can't remember, I think that was him and I, we came up with cleavage-resistant RIP1-associated periodic syndrome. And Dan is like, whoa, nope, I am not saying to my patient that they have crap syndrome. So we had to come up with another name that I really like. And now we're calling this disease cleavage-resistant RIP1-induced autoinflammatory syndrome, and I will call it CRIA syndrome. But we were convinced because we were biased, but we still needed to determine whether really this mutation was driving the disease. And as I said, we had this opportunity of our mice. And when Seth came to us, I was excited. I went to the mouse room and I touched the front head of my mice to see if they had fever. And then I couldn't see it, but that was stupid because the mice, they do not develop fever. So I monitored the mice for quite a while, up to 1 year, and they did not develop any overt inflammatory phenotype or any of the symptoms that were presented by, that were affecting the patient. And here it's an example of a really, really healthy spleen from 3 months old heterozygote RIP1, uncleavable RIP1. But our mice, they are housed in super, super clean facility because our tech are amazing. And the thing is that this is great to be in a clean facility to be able to determine what is the effect of one parameter to X, Y, Z processes. But when you want to really have the real scenario that is happening in human, which is we are always challenged by microbiome product. Our microbiota change if we eat a burger or a yogurt and so on. So what I thought here is that I would challenge the mice with an inflammatory phenotype and see how they would respond and compare to the wild type. So I've chosen LPS, which is a bacterial product, and this is known to be inducing a cytokine storm. And you can appreciate that the mice that were treated with LPS, the heterozygote had a tendency to produce more inflammatory cytokine, and in particular TNF and IL-1 beta. And we did not see much difference for the IL-6 in that case. And the great thing was that here it also took Adam's PBMCs and also treated with LPS, and he also found this exacerbated production of cytokine upon LPS. And we also used another type of microbial product, which is poly-IC, which mimic virus activation or infection. And similar to LPS treatment, we found that we had more TNF produced in the mice serum as well as in the patient of the PBMCs of the patient. So now what's happening is that we think that the CRIA patient, they have an exacerbated response to inflammatory signals. And so Stephen has done his homework. As I said, he was reading a lot about RIPK1 cleavage, and at the time there was not that many papers, and there was those two from 20 years ago that were suggesting that the cleavage of RIPK1 is to inhibit NF-kappa B. And therefore, one would imagine that if RIPK1 is no longer cleaved, there is hyperactivation of NF-kappa B and therefore more inflammation. So what he looked at the P65 nuclear translocation, which is NF-kappa B marker, activation of NF-kappa B. And he found that there was potentially some sort of increase here at this concentration, but it turned out that it was not dose dependent. So we were not sure, we were not thinking that the NF-kappa B was defectious in the CRIA syndrome, in the uncleavable RIP1 heterozygote patient. And we have confirmed that in the mice, that in our cells, when we compare the wild-type RIP1 and the heterozygote uncleavable RIP1, and we looked at different markers of the NF-kappa B as well as the MAP kinase, and we did not see a significant difference between the wild-type and the heterozygote. So that was telling us that it's not a defect in NF-kappa B that was driving this exacerbated cytokine production upon LPS and therefore in the CRIA syndrome. So we went back to our original finding in the homozygote, where we find that those cells were hypersensitive to TNF only. And also because the embryo was rescued if we inhibited both arms, necroptosis and apoptosis. And we treated our heterozygote cells with TNF, and it turned out that they were not sensitive to TNF. It was only when we tickled the TNF signaling, when we just inhibited any of these regulatory mechanisms that was blocking RIP1 to go to that complex. So here I'm showing an example with cells treated with TNF and SMAC mimetic. So RIPK1 is no longer ubiquitinated. There is a rapid formation of that complex, and half of the RIP1 here cannot be cleaved. So you can appreciate that the wild type, the heterozygote were rapidly sensitive to TNF plus SMAC mimetic, and that was the same with the homozygote. And as in our homozygote result, we found that the inhibition of caspases was not inhibiting the cell death, neither in the heterozygote and the homozygote. And like in the homozygotes, loss of RIP3 was nicely rescuing this cell death, or at least bringing it back to the wild type level. And similarly with the inhibition of the kinase activity of RIPK1. So that was all going to the same the same direction, and what we found in the homozygotes. And I just wanted to precise that whether we were inhibiting the ubiquitination of RIP1 or phosphorylation by using IKK inhibitor or MK2 inhibitor, we found the same hypersensitivity to TNF. And also we found that in multiple type of cells, and which was telling us that the entire body, not the entire body, but a lot of the cell type were hyper-responsive to the TNF, and that's probably the reason why the CRIA patient, they were showing those those symptoms. But we had to prove that in vivo, what the contribution of cell death in driving the cytokine, and we went back to our LPS challenge experiment, and here we have taken the uncleavable heterozygote RIP1, where RIP3 and caspase were absent, and you can see that the cytokine level after LPS was back to wild type when compared to the heterozygote. So that was telling us that cell death was contributing to this inflammation in our cells and potentially in the patient. So our model is, so there is few data I haven't showed, but what we think is happening is that uncleavable RIP1 is stabilizing this complex, this famous complex 2, and it brings more caspase, it brings RIP3, which bring more RIP1 and more caspase, and that's why you have this overwhelming activation of cell death. And this fits to our finding on the role of RIP kinase 3 kinase activity versus scaffold function. Because when we inhibited the kinase activity, we did not inhibit necroptosis. It's only when RIP3 was absent that we had a complete rescue of the cell death and a rescue to E12 of our embryo. So we are highlighting a scaffold function of RIP3 in regulating apoptosis here. And what we're showing is that this cleavage is actually in the homozygote, bypassing all the other regulatory mechanism that is on RIP1, which are the phosphorylation and the ubiquitination that are normally keeping in check RIPK1 and stopping RIPK1 to kill the cells. However, in the heterozygotes, we had to inhibit ubiquitination or phosphorylation to be happening. And we think that there is a similar molecular platform that is formed, but in that case, there is non-cleavable RIP1, and the wild-type RIP1 can be cleaved. So the stoichiometry may be different, and if we inhibit these inhibitory regulatory mechanism, that's where we're driving and pushing into cell death. And one would think that when do those patients have SMAC mimetic? Do they have SMAC mimetic for their breakfast or something? And the obvious answer is no. But one scenario that can happen where the IAP level can be low is during an inflammatory processes. Because in actual fact, during inflammation, you have different signaling pathways that are activated. And all those signaling pathways, they all work together. And if you are not sick, that's where you have an optimal inflammation happening and you don't feel it. But there are other receptors that naturally induce the degradation of CRPs. So maybe in those patients, the fact that they have after the RIP1 that is not cleaved, this is pushing, when IAP are degraded by those receptors, pushing to activate this molecular platform. So coming back to the beginning of my talk, does cell death contribute to inflammatory disease, and does cell death contribute to the CRIA syndrome? And I think we've clearly showed that necroptosis is not involved in at least the lethality of the E10. And here I'm showing really preliminary results with the heterozygote RIP1 crossed to MLKL and challenged with LPS, and you can see that they responded like the straight heterozygote, telling us that necroptosis might not contribute to the CRIA syndrome. So if it's not necroptosis, is it apoptosis? And I mentioned before in the beginning of my talk that apoptosis is considered as immunosilent. So what we can put forward, though, is that this molecular platform is actually having its own transcriptional response that is nothing to do with activation of caspases or even necroptosis. And one of the candidates, and the one that I preferred, and I'd like to pursue on that, is the role of RIPK1 in this potential transcriptional response that would induce cytokine and induce CRIA syndrome. And the reason I'm saying that is because now we know that this kinase activity is not solely to inhibit necroptosis, but we're showing that it is also regulating caspases. And in actual fact, with a postdoc that I was having with Devo, Nima, and Jared Sandoz, we have done some phosphoproteomic in the kinase dead cells. And what we found is upon TNF, when RIPK1 kinase activity is inhibited, we found 193 protein that were differentially phosphorylated. So this result was telling us that the kinase activity has other role than activating necroptosis, and maybe these protein are implicated in activating a transcriptional response to induce cytokine. And the other candidate is caspasate, and the reason is from the fact that, again, apoptosis immunosilent, but there is a number of work that has talked about the non-apoptotic and non-necroptotic role of caspasate, and in particular his scaffold function. But to us, to understand whether apoptosis is driving the cytokine, one way would be actually to cross the mice to the latest effector of apoptosis and leaving caspasate intact. I just want to finish with 2 slides about the therapeutic avenue of the patient. So one would think that the kinase activity would be the one that you would target. So using a RIP1 inhibitor. And this is great because they are under development. But there is a long way before those patients can get those drugs if they go to clinical trial and so on. And I was thinking a lot about TNF and how those cells are sensitive to TNF killing. And obviously, one would think that if you use the TNF, you would be able to completely cure those patients. However, none of the patients responded to TNF therapy, and neither to another pro-inflammatory cytokine, which is IL-1. So all the patients that tested IL-6 had a remarkable response to it. So now the question is it because RIP1 is involved in the IL-6 signaling, or is it because TNF has a transient role activity and there is a persistence of the IL-6 expression? So another cross, sorry Tobias, there is more genotyping to do. So this is ongoing, and we will determine whether RIP1 is regulating the IL-6 signaling. So I'd like to finish to thank all the people that helped me for this fantastic journey. As John said, I was not sleeping a lot last year, and so I would like to apologize if I was a bit weird and if I was a bit... especially to all the people that help us to determine and validate this new disease, and to highlight all those new questions in the cell death field. And so, again, Dan and Stephen and Hiro, that was fantastic, and they were extremely generous. It was really easy collaboration with them. We were all excited about it. And Magic Lab, Andrew, I have more mice to make, so I'll come to you. But that was really, really nice to have this next door. It was extremely handy. At We High, as I mentioned, it was really, each floor you go and you have an expert in everything that you want to address. So this is fantastic. And so Anna and Christine, as you know, they've helped me on determining what was happening at E10. Christine, she taught me how to dissect E10. And Anna, she dissects in 32 seconds, 12 liters. So I was quite amazed, and with her, within 3 liters, we knew what was happening. So Christy, really nice adventure, and she was really nice to give me a lot of access to that machine. Seth, the mediator, the informator, thank you for that, and also his input in auto-inflammatory disease, because he himself discovered one that is driven by IL-1 mutation. And I haven't talked about the work that we've done with Ed's team, Mark, Amanda. We have looked at T cell and B cell and what was this uncleavable RIP1 was doing. Kate, she was fantastic for all these LPS treatment. She was coming from the Hudson to help me and to explain me what would be the best timing to look for the cytokine and help me harvesting the mice. Manolis in Germany, it was amazing because he was making the mice, and both of us, we were in parallel with Lawrence analyzing the mice, and it was really great because it was comforting in me in the result I was getting. And the Silk Lab, and in particular, the amazing people that were able to support me while I was not sleeping. And a big thank to Deep Xiao. She has followed me for the last couple of years, and she left just before we published, but she actually produced all the preliminary results of this study. All of them, that's her. And Tobias joined the board after she left me, and the same, he have repeated all Deep's experiments. No, it was an amazing help, and I did not give him a minute to breathe for a couple of months, and I'm really thankful for his patience. Lynn, amazing PhD student. She was here at 10 p.m. checking on me if I needed help, and this is something that from a PhD you would not expect. Nima, as I mentioned, he produced some phosphodata, but he helped also determining what was happening in the cells. And Holly, she has helped in analyzing the E10 vascularization defect. And Natasha, she was amazing as well in making a lot of cloning for us. And the last but not the least, John, for the fact that he was coming on Sunday to do some cloning.
Thank you.
And sorry for going over time.
So we have really gone over time. You all know Najoua. I'm sure you won't be shy to approach her with your question. Is there perhaps just one burning question that somebody cannot keep to themselves, or shall we let them go? No? You're joking? No? Ashley?
You think the cellular mediator of this inflammation?
Sorry?
The cellular mediator of the inflammation.
We don't know. I've done reconstitution where I looked whether there is the immune compartment or non-immune compartment, and in both cases we have this hyper exacerbated response. So we don't know. Yeah.
Okay, as I said, please don't be shy. Ask Najoua questions anytime you like. And thank you all. Have a great afternoon.